Good afternoon. It's a great pleasure to be here. Thanks for the invitation to speak at the ENAC Research Day. So I'm going to switch gears, as expected, and talk about the big problem that faces environmental microbiology, which is finding microbes. Well, isolating microbes. We'll get to that. And this is what I call finding a needle in a haystack. I will tell you what the needle is and what the haystack is in a few moments. But first, let's get on the same page. What's a microbe? Okay, microbe is a microorganism, so we shorten microbes. The bacteria, archaea, microalgae, fungi, and protists, and also viruses, which I didn't put here. And what makes microbes microbes is that they're small, so you can't see them with the naked eye. You need a microscope. And microbes tend to live together in what's called a microbiome. You've heard the word microbiome, I'm sure. It's quite prominent in the popular press. And what is a microbiome? Microbiome is just an assemblage to basically all the organisms, the microbes that live in a specific environment. And what's important to know about microbiomes is that they're very diverse. So there's a lot of different microbes that are present in a single microbiome. So it's a complex assemblage of microbes. And this is the haystack. Also, if you walk out of this talk with just one concept, I would like it to be that microbes are everywhere. They're on our skin, they're in the ocean, they're in the soil, etc. So I'm just going to give you a few examples of microorganisms, or microbiomes, of where we can find microorganisms. So the ocean microbiome is a big one. Very important. You have microbes in the water column, such as you've heard of plankton, right? There's also microbes that live in the ocean floor that can withstand high pressures, for example. And one of the very important processes that is catalyzed by this microbe is the CO2 consumption. So the removal of CO2 from the atmosphere, the formation of biomass, and then this biomass settles to the bottom of the ocean, and that's a way of removing CO2 from the atmosphere, a natural way of removing CO2. Another big microbiome that's very important is the soil microbiome. It is the most complex microbiome on this planet. In 1 gram of soil, you have 10 to the 9, 1 billion to 10 billion microbes. And they play a very critical role in soil health and, of course, in agriculture and plant growth. And also for sequestration of carbon from the atmosphere. And finally, one that I don't study anymore, but you've heard of the human microbiome, we basically have more microbial cells than we have human cells in our body. I'll leave it at that. But those are the sort of standard microbiomes you might have heard of. How about the microbes that live in extreme environments? In the cryosphere. There are algae, microalgae, there are bacteria and archaea that live in snow, ice, and the permafrost. These are called psychrophilic microbes. And just to show you an example, red snow, you've probably seen this in the Alps, are microalgae that live in the snow in the spring. There's also high temperature environments where microbes like to live. Some microbes like to live. For example, Yellowstone, you see a nice picture here of Yellowstone. These colorful hot springs with different colors. These are different organisms, different archaea that are adapted to different temperatures. And finally, hypersaline environments. So these are environments where there's high concentrations of salt. Here, of course, I select this image for its impact. Colorful, beautiful. And these are salt ponds in San Francisco, near San Francisco. They just fill up the ponds with water from the ocean, from the bay. And then as water evaporates, the salinity increases. And because of these ponds are in different states of evaporation, you have different archaea that are growing in them, and each has its own pigment. And so that's why you have these, different colors. Just as a little tidbit, the most extreme halophile, a salt-loving organism, can live in up to 30% salt. That means 300 grams of table salt in 1 liter of water. You can try it at home and see how salty that is. Okay, well, this basically just gives you a little overview of microbes and how they're amazing and can live in many different environments. But why do I care? I care because we can use microbes as engineers. For example, for the degradation of contaminants. Here I show an example of a contamination by perchloroethylene very toxic contaminant. Many sites are contaminated with this organism, this contaminant. But there are microorganisms that can degrade that in situ, in the groundwater, and convert into a harmless form. Same thing with some metals. Chromium can be remediated by microorganisms. And also in the context of global change or climate change, organisms are increasingly harnessed for their ability to transform chemicals into useful chemicals. So for example, fixing CO2, cyanobacteria can do this, and then the product of that carbon fixation can then be further transformed to usable feedstocks for the chemical industry to avoid using fossil fuels. So I hope I've convinced you that microbes hold tremendous potential. They have the ability to transform chemicals. They're basically little chemical factories. They can degrade toxic chemicals. They can produce novel chemicals that are of high importance. They are also adapted to extreme conditions, so they can do things under conditions that most other, the extreme conditions such as high temperature or low temperature, high pH or low pH, high salinity, high pressure, and this has tremendous potential for industrial applications. But, where's the but? The but is that many of these organisms that we would like to be able to use for all these applications are not readily grown in the lab. So you have your microbiome, that's the haystack. We want the needle. We want that one organism that can degrade that toxic compound, that can make that usable chemical. And that, herein lies the problem. They don't grow where we want them, in the lab like we want them to do. And what I'm showing here is a portion of the tree of life. You don't have, it's a lot of colors. The main thing you need to look at here is that the dots. You see there are a lot of red dots. Each one of these little, limbs of the tree represents a group of microorganisms, and the ones that have the red dot next to them are those that cannot be grown in the lab currently, for which we don't have an isolate, a pure organism that we can work with in the lab. So entire branches, you can see this entire branch here, has basically not a single organism that we can work with in the lab. This is a problem if we want to develop the potential of these organisms. So we would like to isolate these organisms, but our isolation tools are limited. This is the standard isolation tool, agar plates. You take some culture, you put it on the agar plate, you spread it around with the spreader, this is coming, and then you wait. You let it sit for say, overnight, and then you get these colonies, these little dots that each represents an original organism that grew into a colony. Then you pick one colony at a time, and you grow it in liquid. As you can see, this is tedious. This is not something that's going to allow us to cultivate a lot of organisms in an efficient manner. The other problem is that this so-called great plate anomaly. So this agar plate that you see, this is our standard way of cultivating organisms. Well, it doesn't work. Only 1 to 5% of existing microbes can be grown this way. So 95 to 99 are just not accessible right now. So these agar plates are convenient, but not very good at doing their job. So we need new techniques. We need new novel tools. We need something that's high throughput, that you don't have to pick one at a time. We need to target specific microbial processes, like a very targeted approach. We want to find a bacterium that can transform cholic acid into deoxycholic acid, that remove the hydroxyl group. That's what we want. We want to be able to pick that one from the assemblage of microorganisms. And we want to be able to target those organisms in that whole part of the tree of life that cannot be accessed, to all the red dots. So we've started working on this in my lab by developing a high throughput isolation technique. So we take some soil, remove the microorganisms from it, and then we use microfluidics where individual capsules, you can see these capsules here, which capsule encapsulates only one organism, and it's made of a liquid core and a hydrogel shell, which is rigid, but semi-permeable, allowing for compounds to come in and out. And then, basically, you can make these, a lot of them, quickly, and allow them to grow. And so you see the ones that are full are ones in which growth has occurred. You can actually even see in this video the live growth of these organisms in the capsules. So they grow. And after they've grown, we need to separate the capsules that have growth in them from those that don't have growth. And so we stain them with a fluorescent dye that's green fluorescing. And then we separate the ones that have fluorescence, the ones that are not fluorescent, and put them in these little microplates, basically little wells. 96 or 384 of these wells. Each one has one of these capsules that has growth in it. And so now we have a whole bunch of isolates. We can do 10, 20, 30 of these plates. We have lots of isolates. But that doesn't help us. That helps us with the high throughput part, but not with the targeted isolation part. So we have to find a way to actually target a specific microbial activity. And here I'm going to give you an example with arsenic. So this is an arsenic molecule, arsenite. And it can be transformed at this enzyme called ArsM. This is not very important, but just that the hydroxyl group becomes a methyl group. Who cares? But it forms something called MMA, monomethylated arsenic. And this MMA is actually not good for rice cultivation. It's a deleterious compound. And it's formed in the soil where the rice is grown. You have this process that's happening, and it's microbial mediated, by the way. So we would like to know more about what's going on there. How is it happening? Can we control it? Because this MMA ends up in the rice grains, and this is not beneficial for the humans, but also it turns out for the rice plants. So what we would like to do, and if you took to look where the methylated arsenic is in the rice grain, this is an XRF of a rice grain, you see it's everywhere. Anyway, the point here is that we would like to isolate these organisms. So we need a way of detecting MMA, this compound here, fast in high throughput manner. So we developed a biosensor. A biosensor in this context is a bacterium that is engineered genetically and is able to respond to the presence of MMA by fluorescing. So here you have the inactive biosensor. It looks like a normal bacterium. In the presence of MMA, it glows green. Yeah. Okay, so practically speaking, how does this look? So if you plate a biosensor, it's just a bacterium, so you can put it in the agar plate like I showed you in the other video. And then you can put a drop of either arsenic, the former kind, the substrate, or the product, and then look at what happens. If you put arsenic, nothing happens. It's just some growth. If you put MMA, you see that there's a fluorescent signal that appears where you have put that drop. So this is how the system works. You can just see light coming forward. So now, in order to be able to use this in high throughput, we need to calibrate this biosensor so we know how much MMA is present when we have a specific signal. So how do we do this? Well, we just measure, basically put the bacteria up there in a bottle with a different concentration of arsenic, and we look at how much light is produced, how much fluorescence. Here at the bottom, you see there's no arsenic, no signal. If you put inorganic arsenic, so the substrate, you see that there's a signal. And then if you put the MMA, the target, you see there's more signal. But if you look closely, the concentrations are very, very different. It's 1,000 times more inorganic arsenics. The blue curves represent 1,000 times more arsenic than the MMA. So what this means is that this biosensor can capture very small concentrations of MMA, and also it's highly specific. It targets one compound, not two compounds. So then, we just combine these two techniques. We have the isolation, which I told you about before, and now we have the biosensor. So we combine these two, we grow the bacteria like we did before in the micro-well plate, but now we add inorganic arsenic so that this process of methylation, this formation of MMA, can happen if it's going to happen. And then we remove, after we let it grow for a while, we remove the water, the aqueous part from these 96 wells, and add the biosensor. And then we scan with this machine that you see here, a multi-plate scanner. And exactly like before, we measure the fluorescence. It's normalized fluorescence. So you see that at the bottom, in blue, we have the signal from the arsenic only, the one you added. So any signal below that is just basically noise. But there we have the positive control, 0.03 micromolar of MMA3. You see it's much higher than the threshold. So anything that happens, any well that registers some signal below the arsenic, the blue line, is just noise. Anything in between, and here I have an example, that could be a potentially MMA-producing microorganisms. And with this process, we were able to isolate new isolates, to find new bacteria that can do this. And so, I've given you this example for arsenic, but the same strategy can apply to other problems. Like, for example, we have a project now that looks at biodegrading explosives in soil. As you may know, there are a number of theaters of war happening in the world at any one time. Now it's particularly bad. And after the war ends, the deminers come in, they take the large pieces of unexploded material, but there's all this explosive is still there, and it's highly toxic, TNT, RDX. And before the soils can be used, they need to be remediated, cleaned up. And so we're looking for microbes that can do this from the soil that's contaminated. Once we have them, you can basically grow them up and apply them to the soil and add nutrients if necessary so that they can sort of fast-track this process of explosive degradation. And the other example is N2O. So N2O is a greenhouse gas. It's naturally produced by microorganisms in the soil. But we'll be looking for an organism that can transform this greenhouse gas into N2, nitrogen, harmless gas. Most of our atmosphere is made up of it. And by doing so, we can mix in this N2O removing organism into the topsoil. So this is for agricultural applications, because agriculture causes the production of a lot of N2O. And so then this N2O will be transformed to N2, which is, as I said, harmless. And this means that less N2O will be produced, will be emitted from the soil. So to conclude, I would like to hopefully, I made the case that microbes offer inexpensive and environmentally friendly approaches to address some of our societal issues, and also that we need new tools, and new tools to isolate and cultivate these organisms are key in order to be able to access all this potential. And with this, I'd just like to acknowledge Hugo Saleh, who's done most of this work, and Karen Maybaum, who has designed the biosensor. And then Luna, Marion, and Matteo are internship students who also helped with this process. And then Manon Frucci, who is a lab technician and who's responsible for some of the videos that I showed. And with this, I'd like to thank you for your attention.